Information and Communication Technology presents Educational Satellite Television Programs.
Hello, teacher. Hello, students. Welcome to today's lesson on protein synthesis. I am sure you recall that we began learning about this topic in our last lesson. We discussed the process of transcription and we learned where it occurs. In today's lesson, we will describe the full process of protein synthesis. We will also discuss the functions of mRNA, tRNA, and rRNA in the process of protein synthesis. Shall we begin? As you already know, protein synthesis is the process by which cells build proteins. While this term can be used simply to describe protein translation, it is more often used to describe the multi-step process that starts with transcription of nuclear DNA into messenger RNA. Previously, we discussed transcription. As a review, please describe what happens during this process. After this activity, we will discuss what happens during protein synthesis after transcription has occurred. You may begin now.
Students, let's get ready. Begin. Time's up. Let's get back to our lesson.
Hello, everyone. Were you able to recall the process of transcription? Transcription is a process that occurs within the nucleus of a cell. A complementary RNA copy of a base sequence of DNA is created. The RNA polymerase binds with a section of the DNA and uses the antisense strand of the DNA as a template for synthesizing the mRNA. The RNA polymerase arranges the free RNA nucleotides into a chain that has a complementary base sequence to the antisense strand of the DNA. The new completed molecule then separates from the DNA, and the DNA recoils itself. Was your answer similar? If so, great work. Let us continue by discussing the rest of the steps involved in the protein synthesis process. After transcription is complete, the mRNA travels from the nucleus through the pores that exist in the nuclear envelope. The mRNA makes its way towards the ribosomes. At the same time, molecules of transfer RNA, or tRNA, carry free amino acids from the cytoplasm to the ribosomes. Once the mRNA and the tRNA reach the ribosome, the process of translation can begin. The ribosome reads the mRNA code and assembles the amino acids carried by tRNA into a protein. The tRNA provides the corresponding necessary amino acids to the appropriate mRNA codon. Ribosomal ribonucleic acid, also known as rRNA, is the RNA part of the ribosome. This organelle is the site at which protein synthesis occurs in all living cells. rRNA provides a mechanism for decoding mRNA into amino acids. It also interacts with tRNA during the process of translation by providing peptidyl transferase activity. Peptidyl transferase activity provides links between adjacent amino acids using tRNA. This all occurs during the translation process. Students, we have learned about mRNA, tRNA, and rRNA. Please explain each term and describe the function of each in the process of protein synthesis. Write your answers on a piece of paper, and I will be back in a few minutes to discuss the answers with you.
Students, let's get ready. Begin. Time's up. Let's get back to our lesson.
Hello again, students. I trust that you did not find this activity to be too difficult. Let us go through each of the terms together and discuss their functions. mRNA stands for messenger ribonucleic acid. It is a nucleic acid that transmits the genetic code from DNA to ribosome. tRNA stands for transfer ribonucleic acid. It transfers individual amino acids during translation. rRNA stands for ribosomal ribonucleic acid. It is the site at which protein synthesis occurs in all living cells. Were you able to define all of the terms correctly? If you had any trouble, please review your notes. Students, now that you have learned all of the steps involved in the process of protein synthesis, as well as the functions of each element that is involved, please draw a diagram depicting protein synthesis. Label each of the parts that we have learned thus far, including DNA, mRNA, tRNA, and rRNA. Go ahead and get started.
Students, let's get ready. Begin. Time's up. Let's get back to our lesson.
Hello again, students. I am sure that all of your diagrams look great. Please take a moment to compare your diagram to the one on the screen. Make sure that you have labeled all of the components correctly. The DNA is located in the nucleus of the cell. This is where transcription occurs, and the mRNA is created. The mRNA and the tRNA then both travel to the rRNA. This is where translation occurs and a protein is created. In today's lesson, we described the process of protein synthesis and the function of messenger RNA, transfer RNA, and ribosomal RNA. We learned that after mRNA is created during transcription, it travels to the ribosome. We learned that free amino acids are carried to the ribosome by molecules of tRNA. At the ribosome, the mRNA is read and amino acids are assembled into a protein. In our next lesson, we will further explain the process of translation. This brings us to the end of our lesson for today. Until next time, thank you, teacher. Thank you, students.